the surface of the disc. The single arrow points to the sample port where the samples will be introduced into the disc. The two arrows pointing towards each other mark the fill line that indicates when there is the right amount of sample in the disc.
Next, you have a piccolo pipette and pipette tips supplied by Abaxis, which are designed to accurately measure and easily transfer the sample from the tube to the reagent disc. These pipettes are easy to use and hold the correct amount of sample required by the reagent disc. Now let's introduce our first sample into a disc. Lithium heparinized whole blood samples should be analyzed within 1 hour of collection. Heparinized serum or plasma should be tested within 5 hours of centrifugation after separation from the red cells.
After extracting 100 microliters of sample using our pre-measured pipette, place the tip of the pipette into the sample collection port marked by 1 arrow. Make sure that you use even pressure and don't allow any sample to re-enter the pipette after it has entered the reagent disc. This could cause an air bubble, resulting in a canceled disc reading. Tilt the disc to 45 degrees with the sample port above the fill line so that the entire sample flows into the sample chamber. Continue to add approximately 100 microliters of sample until the sample forms a straight line between the two arrows showing the fill line. Remove any sample spilled on the outside of the disc with a lint-free tissue, taking care the tissue does not withdraw any sample from the sample port. Once the sample is in the disc, it must be inserted into the analyzer within 10 minutes. Press Analyze on the touchscreen to open the disc drawer. Place the disc in the recessed area in the drawer. Make sure it is seated properly. Press Close on the touchscreen. The analyzer then closes the drawer. The information you enter from this point on depends on how you configured your piccolo. See your operator's manual for details. Once the disc is fully loaded into the analyzer, follow the on-screen prompts to enter the appropriate data. Remember, the right arrow key functions as a dash and the left arrow key functions as a backspace. When finished, press Done.
At this point, the Piccolo Express takes over. The analyzer checks the disc type, lot number, and expiration date, and then begins processing the sample with no further input as soon as the drawer closes. If the disc is not found to be the correct type or is expired, an error message appears. Repeat the analysis with another disc of the correct type. While the sample is being analyzed, its progress can be monitored by the status bar at the top of the touchscreen.
Sometimes it may be necessary to cancel an analysis. To cancel an analysis in progress, press Cancel on the touchscreen. To confirm cancellation, press Cancel again. The analysis is then canceled and the disc drawer opens. Remove the disc from the drawer. The analyzer is now ready to perform another analysis. Press Close to close the drawer. When the sample is finished processing, the Piccolo Express stores the results and notifies you that the analysis is complete. By default, the analyzer automatically prints the results of the analysis. After printing the results, press Open to open the disc drawer. You can now remove the reagent disc from the drawer and discard the disc in the appropriate biohazard waste receptacle. The Piccolo is now ready to analyze another disc. To analyze another sample, insert the new reagent disc and repeat the same procedure. The results printout has an adhesive back so that it can be easily attached to the patient's file. At the top of the results printout are the reagent disc type, date, time, sample type, sample ID number, gender, disc lot number, and analyzer serial number. The test results are printed in 4 columns. The first or left column lists the chemistry names. The second column contains the analyte concentration for each chemistry. The third column contains reference ranges, and the fourth column shows their specified units. Results outside the reference range are indicated in the results by an asterisk printed next to the analyte concentration. Results outside the dynamic range are indicated in the results by a less than symbol printed next to the lowest value of the dynamic range, or a greater than symbol printed next to the highest value of the dynamic range. 2 lines appear on the bottom showing QC and sample integrity. The message QC OK is printed on the results when results from these reagents are within the expected ranges. Sample integrity is indicated by HEM for hemolysis, LIP for lipemia, or ICT for icterus. These indices indicate the degree of hemolysis, icterus, and lipemia found in the sample, measured on a scale of 0, clear, 1 plus, slight, 2 plus, moderate, and 3 plus, gross.
The Piccolo performs a series of internal quality checks to ensure accurate results. Intelligent quality control is designed to detect any abnormality during the complete analytical process and ensures no erroneous result is reported. Refer to pages in your Piccolo Express Operator's Manual for a thorough listing of common errors. Sometimes, due to operator or analyzer error, improper sample collection, sample contamination, or other reasons, the Piccolo IQC will detect an abnormal condition and cancel the test. This will be indicated on the VGA screen. When the analyzer detects a problem, it can suppress certain chemistry results. When this occurs, the analyzer prints troubleshooting flags and prompts you to print or transmit an error report. When this happens, you should discard the disc in the appropriate biohazard waste receptacle and run the test again using a new disc and fresh sample. If the test cancels a second time, follow the on-screen prompts and print out an error report.
The Piccolo automatically stores IQC and troubleshooting data. Troubleshooting data can be printed or transmitted to a computer. To recall and print troubleshooting data, press the folder icon on the analyzer screen. The following selection will be displayed: Last Disk, Search, Browse, Transmit All. By selecting the Last Disk button, the results for the last disk run will be displayed. Search allows you to search the stored results by sample ID, date run, patient results, or control results. Browse allows you to browse all the stored tests. Once you locate the result you want, press the Print button. Now you can select to print or transmit, or both, any or all of the available reports for the sample selected. For troubleshooting data, select Error Report. Once printed, press the Home screen to return to the Analyze screen to test the next sample, or the Back button to go to the previous screen. Then call 1-800-822-2947 and speak with Anabaxis Technical Services representative and provide them with the printout data. They will instruct you on what corrective action needs to be taken. Aside from the printed results, test results are also stored in the Piccolo. The Piccolo Express contains enough memory to store up to 5,000 test results and controls. These results can be recalled and printed at any time the disk drawer is closed and a test is not running. To recall the results of the last disk run, press the Recall icon located on the home screen. As an example, select and press Last Disk. The results from the last disk are now available for review. Use the up and down arrow keys to scroll through the results. To print or transmit your last disk run, press Print. Then select Print and/or Transmit. You can then select the following reports: All, Results, IQC, or Error Report. Then press Results. The display briefly shows Sending Report while the report or results are being printed or transmitted. Then returns to Select Report. You can return to Home or Back to retrieve more results. If the disk produced no results, only the Error Report option will be available. Remember, you can also use the Recall function to search for a particular results record according to its patient ID number or date. You can also view a list of all patient results or control results sorted by date. For further instruction on these recall functions, refer to your operator's manual.
That pretty much sums up how to run a test using the Piccolo Express. You can always refer to your Piccolo Express Operator's Manual for more detailed instructions. Piccolo Express runs waved and non-waved panels. It is critical to establish IQC and verify accuracy, precision, and reportable range when running non-waved panels prior to reporting patient results. These procedures follow the Centers for Medicare and Medicaid Services and CLIA regulations to meet non-waved complexity certification. Abaxis provides these instructions during setup and installation of your Piccolo Express. Upon completion of these procedures, you will have successfully established IQC, which allows you to run external liquid controls only once every 30 days, and verified accuracy and precision